So as Sarah mentioned, my main research topic is around double haploid technology, its optimization and utilization. And I was wrestling a little bit with the question how it fits to the theme, but I think I can argue and make the case that DH technology helps with making breeding of resilient crops more efficient. And I want to demonstrate that by using the genetic gain equation. Double haploids are known to speed up the breeding process by developing inbred lines in shorter time, in only 2 generations, as compared to 6 to 8 generations typically. That helps with the denominator of the equation. But in addition, double haploids also help to increase to maximize genetic variance compared to segregating families, and they help with the phenotyping, increasing the heritability. So also the numerator is impacted by the H technology. Before I will talk about the different tasks that we are working on in terms of inducer development and haploid genome doubling in more detail, I want to make the case again that progress in reproductive biology, more generally speaking, has the potential to really help with making step changes in plant breeding. One example that I pulled out is the transfer of rice, which has been bred as line varieties in the past and more recently as hybrid varieties. That there's an example that you can use just 4 genes to convert heterozygous genotypes into apomictic genotypes that can be clonally seed propagated. And one of the genes actually came out of the DH process. Matrilineal was just isolated in 2017, and combining it with 3 other genes helped to convert rice into an apomict. And that's a model for other crops. Another important transition, of course, from one variety type to another is in maize, from originally population varieties, which had fairly low yields, into hybrid varieties. And here, of course, recurrent selection, quantitative genetics were most crucial, and Dr. Hallauer was a main player in this connection. He's in the audience. Double haploids have been around as a concept since 1940s, 50s. They were introduced by Sherrod Chase, who was here at Iowa State. He came up with the concept in 1953. Actually, he had the first DH lines available, so 70 years ago. However, it took about 50 years until those double haploids and the DH technology was really implemented into major breeding programs. And today, I would argue that double haploids and genomic selection are crucial to help maintaining the steep annual genetic gains that we are seeing. So they are important routine technologies in major breeding programs, and they produce hundreds of thousands of DH lines every year. However, not every breeding program has access. There are many smaller breeding programs, international programs, public programs that still benefit from getting access to DH technology. And even for the major breeding programs, it's a fairly costly process. So anything that can help to optimize the process can help with the economy of the process. It's shown how it works. In maize, sorry. So in maize, the maternal haploid induction process is used. That means you use a donor as female and the inducer as a male. Haploid lines are extracted from the female, and for that reason, it's maternal haploid induction. You need a genotype, a specific genotype, the inducer, to result in about 10% offspring that only have the maternal genome. To identify those, by default, the RNJ, an anthocyanin gene, is used to help by coloration to identify kernels that carry this haploid seedling or germ. You can see an example here. Kernels are colorated and the germ is reddish, and you would select kernels that have no coloration here. Those kernels are then subjected to a treatment with, by default in most cases, colchicine to help with doubling the genome to obtain DH plants. In most cases, these plants are chimeric. They contain haploid and diploid cells side by side, and you need the diploid cells to result into the floral organs to be able to self-pollinate. So it's an inefficient process which succeeds in maybe 10 to 20% of the cases. Also, if you have to treat seedlings, then you have to transplant them instead of sowing. So the steps where the process can be made more efficient is in inducer development or in the doubling process. And here specifically, we were interested in the question whether there's genetics that could help so that we don't need to use any application of chemicals any longer. Inducer development has some history. As I mentioned, it started by Sherrod Chase in the 1940s, but the first inducer first became available in the early 1960s by Ed Coe from Missouri. And that stock 6 is really the ancestor of all modern inducers. This is only a portion of inducers that is available globally. There are many more today in all regions of the world. We obtained access to an inducer from University of Hohenheim, from Professor Geiger, and started working with DHs in collaboration with the Plant Introduction Station in their germplasm enhancement of maize program. That resulted in GEM DH lines, which have been made available and are still used. But at the same time, we decided that starting a DH facility may be valuable to public and smaller breeding programs. And that's what we did in 2010. But we also learned quickly that the inducer that we got from Germany wasn't really well adapted. So it was typically susceptible to storms, lodging, and it was still viable. We could collect the pollen from the ground, but we decided we need something that is standing up under IO conditions. Also, seed production was a problem. So these were the two main traits that were making us develop adapted inducers. But once we started the process, then of course we also started to look into increasing induction rate and to look into other markers or other traits that could be beneficial. One of those is oil content as a means to help with sorting the about 10% haploids from the remaining 90% heterozygous genotypes that you don't want. The concept was coming up from Professor Chen in Beijing, but really Professor Melchinger in Hohenheim, he was pushing this concept further ahead. High oil inducers became available there. And the main point is that you would like to have heterozygous offspring that have an oil content that is clearly higher than that of the haploids, which are derived from the maternal genotypes. Typically, field corn has 3 to 4% oil content, and you need inducers that have 10% or higher oil content so that the heterozygous offspring have hopefully 6, 7% oil content. If that is successful, then you can start using machines and automate the sorting process. This is something which is going on currently. So we have the Q sorter and the single kernel NRS equipment available on campus, and hope that those will eventually help us with the automated sorting instead of the visual manual sorting. You can also use image analysis potentially, because higher oil content also leads to increased embryo size. Another case for developing specialty inducers is popcorn. So when we started with the DH facility, we got contacted by popcorn breeders, and we realized that selecting haploids is fairly difficult with these small kernels. Unfortunately, you can't just pop the kernels and then kind of select, keep the haploids and eat the other ones or similar. So we decided we needed another marker system and incorporated red root marker based on PL1, which is independent of the R and J gene. In addition, most popcorn breeders use cross-incompatibility, which is mediated by GA1. So the dominant GA1 allele prevents pollination by transgenic field corn, which usually is small GA1 and cannot pollinate popcorn. It cannot overcome this cross-incompatibility barrier. In order to produce DH lines in popcorn, we therefore developed inducers that are carrying the GA1, large GA1 allele. Be sure with the Okay, so this is now about haploid induction rates. This is a photo from Euro Chen in the audience. It's a laborious trait. If you want to determine induction rates, you have to do many hand pollinations. For each line that you want to test, you need to do about 10 test pollinations. We try to get about 1,000 kernels to be able to accurately determine the percentage of haploids in the seed obtained. And for that reason, we thought that this is a good trait to consider genomic selection. We did an initial study with the first set of inducer families that we had available, maybe 6-8 years into the DH facility, about 160 families. And we were genotyping those. We had the phenotypic data, and there were also some public inducer lines available, and we asked whether genomic selection is promising. To test that, among others, we were asking in cross-validation experiments, what is the prediction accuracy of haploid induction rate compared to other heritable traits, highly heritable traits like plant height, and it looked like that haploid induction rate is highly heritable. So we felt confident that this would make sense and is promising to move forward on. We also intended to use genomic selection and combine it with availability of DH lines. As I mentioned before, this combination is particularly powerful. But ironically, if you try to develop DH lines for inducers, it's prevented by the fact that we already have the color gene fixed in the inducers if you use them on the donor side. If you use an inducer breeding population, this white embryo would be colored, and we couldn't use our existing inducers to make induction crosses and to identify haploids based on lack of coloration. We made use of the fact that we knew some germplasm is difficult to work with. So we learned that Flint germplasm is usually suppressing the coloration of kernels. And we also knew that this is in most cases caused by the C1i gene. And we capitalized on this fact by developing an inducer that now carries C1i instead of R and J, so that we could use an inducer to extract and identify haploids in our inducer breeding population, which has the coloration, and we would just invert the coloration or the color selection scheme by looking for kernels that now have a colored embryo, whereas the heterozygotes would have no coloration. And this worked. Uru has been able to develop about 400 inducer DH lines, which he then characterized. About 200 of them he determined induction rates. We did find transgression for haploid induction rate. We found inducer DH lines that exceeded 20% induction rate, and that will be the basis for attempting to do genomic selection. He has all data ready. He will finish this summer, so he will be able to tell us in his defense how well genomic selection works. And we already use this information for parent selection in the next PhD project by Wenke Grüning, who will evaluate whether parent selection based on genomic selection works. Once these data are available for genomic selection, we can also use them in the GWAS framework and try to identify candidate genes involved in the induction process. So this is where we are standing right now with regard to our inducer development activities. In blue are the inducers that are available. Midwest inducer with 10 to 15% induction rates. I mentioned the GA1 inducer, C inducer. With a first version high oil inducer available, we think that we can get inducers with 10 to 15%, 15 to 20% induction rate from Uru's work, and I believe it will be possible to exceed 20%. Question is, where is the limit here? We will maybe find it out in Wenke's work or just continue after that. And there's some other specialty inducers that we are working on. With that, I'm now leaving the inducer side, and go to the donor side. What we learned in this DH process is that there's a genetic response of the donor to both the induction process, but also in terms of the genome doubling. For example, using the same inducer, if you induce a wide range of donor genotypes, you can find genotypes that are almost recalcitrant. You hardly find any haploids, whereas other genotypes have maybe 20 or more % of haploids. This variation we studied further in genetic and marker-based analysis. We found there's some QTL involved, there's genetics involved, but no major genes. We also felt that this is something that breeding companies or breeding programs probably inadvertently select for, and we didn't give priority to this trait. So we believe that over time, commercial breeding programs will increase their induction rate because there's selection ongoing in their elite materials. So we focused on the genome doubling side of this aspect, where we also observed genetic variation. And we are highly motivated to do so because I mentioned the transplanting. These haploid plants are very frail. We have to make sure they are really brought into close contact with soil, otherwise they dry out immediately. This is Anderson, PhD student who did work on SHGD, but it could be any other person, including myself. So we are really wanting to avoid this greenhouse planting, colchicine treatment, transplanting step, and if possible, select haploids and go directly into the field. So the question was, can we find genotypes that have a substantial contribution to male fertility, and in particular haploid male fertility? From literature, it was known that there's still a certain level of haploid female fertility. If you pollinate haploids with pollen from diploid plants, you get seed in more than 50% of the plants, whereas the male side typically is sterile. For that reason, we applied a simple scoring procedure looking for pollen shed or for fertile anthers to characterize haploid lines that were derived after inducing inbred lines. So we had haploid lines in the field that we could put out in replicated trials. And we confirmed that the majority of genotypes were actually sterile, and only a few of them had a decent haploid male fertility, and in particular A427 turned out to be a very interesting genotype with a haploid male fertility exceeding 50%. We then moved on in follow-up PhD studies to do biparental QTL mapping. That revealed that there's a major QTL on chromosome 5 near the centromere of chromosome 5. And Anderson Bersignazzi, he was doing a case control study. He used the BS39 population which Arnold Hallower has been adapting to Iowa photoperiod conditions. He extracted DH lines using the colchicine method, and he also crossed a sample of this population with A427 to then derive DH lines with the spontaneous doubling mechanism without any colchicine treatment. And when conducting a case control study, he showed that chromosome 5 is again showing up, which means in lines that were derived in this sample without colchicine treatment, they had this chromosome 5 region from A427 more or less fixed. So it shows it works in A427, but also in different genetic backgrounds. Of course, we then intended to go after the gene using map-based gene isolation. We obtained a high-quality genome sequence using services provided by Corteva. We did a large mapping study, so we started out with 10,000 BC1s, looked for recombinants in this chromosome 5 region, saved recombinants and induced those so that we can do replicated trials again of these haploid families. And we were able to find recombinants, but not in a region of about 20 megabase size. So that's why I call this genomic North Pole. It's not only a cold spot, but it's really, there's nothing happening. There's no crossover whatsoever. And we are continuing to work on this. So you who's in the audience, she will attempt different other methods to try to get at the genes in this region. But at the same time, we can, of course, use the sequence information to try to look for candidates and use a candidate gene approach to maybe get a shortcut. One of them is BMF2 that I will mention a little later. And we were lucky to get a FFAR project funded in 2020. The idea here is that we use mapping studies like the one that I just showed to identify candidate genes. We would then use the DH system in Arabidopsis thaliana, which has been established in 2010 by UC Davis, and combining it with the rich mutant collection to evaluate plants for haploid fertility in Arabidopsis, with the hope to identify genes that have an effect in Arabidopsis, that we could test them subsequently in crops of interest like maize using mutants or transformation. And the idea was that perhaps there are conserved mechanisms like meiosis, mitosis involved, so that the species transfer across a wide range of relationship is possible. We also worked with engineers here to help with scoring for fertile haploid plants. So far, we screened 30 mutants. We promised to do 60 and should be done by this summer. And we use the nice feature that the control genotype Columbia 0 has some level of haploid fertility. So we can identify deviations in both directions. Mutations that lead to a significant decrease are detectable, and we found a couple of those. And we also did find mutants that led to an increase in haploid fertility when mutated. And one of those that led to a decrease are genes like BMF2. BMF2 is a member of the spindle assembly checkpoint complex, the SAC complex, and that is involved in timing when genomes in mitotic divisions are transferred to daughter cells. And one could imagine that if you consider haploid cells that are dividing, and if there's something perturbed in when a genome is transferred to dividing daughter cells, that you could end up with a daughter cell that obtains two genomes. So we think it makes sense biologically, and he did find that a mutation in BMF2 actually did lead to a significant decrease in haploid fertility. He tested that, so Siddiq Abu Bakr, I should not say he, Siddiq Abu Bakr, did this work, and he found a significant decrease with different alleles of BMF2, and he extended that work to other genes in the subcomplex and could confirm that as well. Having that screening pipeline in place, then it enables also to just look into literature. Are there maybe other candidates that could be interesting? And Siddiq has been successful and hitting the jackpot in finding that parallel spindle genes, and there are two versions. One is parallel spindle, the other one is JSON. If mutated, can actually increase haploid male fertility. So that was a hypothesis that Siddiq had after a literature study, which he could then easily test. And he not only found that it increases haploid fertility, but that it's haploid male fertility. He found that by just counting the pollen in these haploids of the mutants compared to the wild type, and the pollen scores in those mutants for JSON or parallel spindle were significantly increased, while on the female side, he was pollinating those haploid plants with pollen from fertile diploid plants and did not see any difference. So again, this haploid fertility is caused by haploid male fertility. And we think it's related to what's going on in these PS mutants, that they are forming diodes in the meiosis process, so only two daughter cells at the end of the meiosis process compared to the usual 4 products. If you start out with a haploid genome, which is replicating, you have ultimately 2 genomes that are divided up to these daughter cells out of the meiosis process. The parallel spindle is helping to divide them up equally to these daughter cells, whereas in the regular meiosis, you would end up with 4 products that only get incomplete numbers of chromosomes. Siddiq has been successful to obtain USDA funding to continue this work in maize. So he has identified homologs in maize, and we ordered already mutants, and this will be evaluated this coming summer. So the conclusion from our work thus far is that there are genetic mechanisms that you can get spontaneous haploid genome doubling, and there are 2 different pathways to get there. One is mutations in the meiosis process, like the parallel spindle genes. The other one is to go through a mitosis or a mitosis process that is perturbed. And we observed that, for example, with the A427 genotype that we started out with in maize. What we're observing there is, if you are looking at leaf sections, that is work that Recep Yavuz is doing, that we see already in younger parts of the plant, in leaves, doubling of cells as measured by the size of stomata. And that has been confirmed by flow cytometry. So this can get haploid plants that early on start to develop diploid cells and may resemble more closely inbred lines that are diploid. We think that comparing these 2, the mitotic pathway may offer advantages of having more vigorous haploid plants. So how do we use this? What are the next steps here? We have an ongoing organic project where Uschi Phi is working with Bill Tracy to use the region on chromosome 5 from A427 to try to integrate it in different backgrounds, in this case in sweet corn. She's using either mark-assisted backcrossing or a procedure where she is doing the phenotypic evaluation of haploid plants in each generation. And so far we are obtaining haploids in subsequent backcrossing steps, and we hope to be able to incorporate it into different sorts of backgrounds. Having haploid fertile plants also opens the possibility to consider an accelerated breeding scheme consisting of 2 generations, where we have diploid plants that are induced, resulting in fertile haploid plants that can then be crossed based on genomic selection to return to diploid plants that are induced again to result in fertile haploid plants, etc., etc. We are currently testing whether this process is feasible and find encouraging results by self-pollinating those fertile haploid plants, also by crossing haploid plants with other haploid plants. So the attempted pollinations that we did last summer resulted in about 50% of successful self-pollinations of these fertile haploids and more than 60% successful crosses on haploid plants. So it seems a path that is possible to pursue, but what we are observing here is that the seed set on these haploid plants is fairly limited. And that leads us to the next question with regard to haploid vigor. The next phase ultimately is to, if you wanted to use this rapid cycling method, doing haploid by haploid crosses, we not only need haploid male fertile plants, but we also need haploid plants that have a decent seed set and thus a decent haploid female fertility. And this is a new area that we are now moving into to try to better understand if there are vigor differences that are genetically determined between haploid genotypes. And so what we are looking at and what we have been done is to re-induce inbred lines so that we can have perfect isogenic pairs of haploid and diploid genotypes. We can then compare them side by side to measure something that we call haploid frailty, which is the reduction in trait performance of the haploid compared to the perfect isogenic inbred genotype. So that is comparable to beta parent heterosis, where you're comparing the hybrid to the beta parent. And the question is whether there's genetic variation for this difference, and if it's genetically determined. We did have initial trials out last summer, and it's repeatable. We had replicated trials, and we found a haploid frailty of 10% on one extreme. That means the haploid is almost the same height in this case as the inbred line, and 50% on the other extreme. That means the haploid is only half the size of the inbred line. And so the question is, if that is genetically determined, can we actually develop and breed for better performing genotypes at the haploid level that at the end would also have an increased seed yield? Looking into speeding up the breeding process with doubled haploids, we have an ongoing project where we try to reach the speed limit ultimately. That's something that Tanner Cook is working on. And here the idea is that with rapid population improvement cycles, they still require sexual plants. So we can use genomic selection, but we still need to go from seed to seed. And the speed is determined by how long it takes to produce seed. The question here is whether it's possible to short circuit this, to go in vitro and then to induce ultimately meiosis in vitro to produce haploid cells that could be genotyped, and then the superior cells could be identified, fused again to return to a diploid stage, etc. So hypothetically, one could get a very rapid cycling in that way, but the grand challenge is to induce meiosis at this time. And Tanner is in the process of developing a screening system together with Zara Kazinia to screen for factors affecting meiosis. So with that, I come to the conclusions. It took a long time to establish the DH process in maize. The main limitation was the availability of effective haploid inducers. That has been overcome. Meanwhile, maize can serve here as a model for other species. Genes have been isolated, and it has already been shown that using this knowledge, it is possible to transfer DH systems in novel species. The next frontier, if you want, is haploid fertility. And could that be used in certain selection schemes? I did want to briefly mention that there's also a particular application that was published by Syngenta in 2019 that the DH process can be used for editing approaches. So you would use the male inducer as carrier of the gene editing machinery. You would capitalize on the male genome being eliminated in the process, but that there's a short set of cell divisions where the female and the male genome would be side by side. A fusion happens. Then the male genome is eliminated after a few generations of mitosis. During this short time, the gene editing machinery can place edits in the female genome. The male genome is eliminated, including the transgenic editing machinery, and the resulting product would be an edited female genome, which hopefully can be doubled. It would be a single generation process to create a mutant in a target gene, and it would be free of transgenes, and because it's pollination based, it would be widely applicable. With this, I would like to acknowledge all the members in my lab that have been contributing, not only the current ones, but of course also the past members. We have current funding from USDA, different projects. I mentioned the FFAR project, and it's particularly noteworthy that there's internal funding, which is really helpful to sustain research over a longer period of time, in particular funding from PSI, Plant Sciences Institute, the Baker Center, and the Fry Chair from the Department of Agronomy. And I would like to particularly thank Uschi Fry, who is really an very important person in running the double haploid facility and also organizing us as a group and the research that we are doing. And we saw such a map before for the seminar series, but I think we can say that over the about 15 years that we have been active, that we were working in different ways, the H-line development, inducer licensing, training students, workshops with various countries or members from different countries, but also in the US. So with that, that's all. Yeah, I think it will very much depend on the species that you're working on. So if you consider soybean, for example, I think getting access to the flowers, being able to work with a species and considering what the constraints are in that particular species with regard to reproduction is really the important point. If a species is self-pollinating or outcrossing, so I'm wondering, for example, if for self-pollinating species, paternal haploid induction would be actually preferable. In maize, we are using maternal induction systems with the inducer as a male, which requires that you have a good seed set. But I'm wondering if you have problems there, if it would be preferable to have, for example, sterile genotypes or use sterility and use then paternal inducers that would be female or used as females.
Thank you, Dr. Lübberstedt. I have a question that I've always wanted to ask you whenever you talk about the oil being used to try to detect that. So that is that the double haploid seed is having a higher oil content, so you can detect that, correct?
So you get about 10% haploids or kernels within haploid embryo and about 90% with a diploid embryo. If you made the cross, you have a field corn with 3 to 4% oil content, and the inducer has maybe 10% oil content. You would only expect to see an increase in oil content in the heterozygous fraction, and you hope that it is kind of intermediate between the field corn and what you have in the inducer. What we are observing, unfortunately, is that if you focus on the per se oil content that you have in the inducer, you may not end up having an oil content in the middle, but it may be closer to what you have in the field corn. So we are learning right now that we have to really not be too focused on the per se oil content in inducer genotypes, but really in the xenia effect, what is transferred to offspring. And that seems to be not closely correlated, which is a bit of a surprise.
Yeah, so just to clarify then, because it was a point I was always confused on, that higher oil is not being transferred then to those DH lines that you're creating, correct?
So they should have the original oil content that was in the donor population. That's okay. Correct, yes.
Perfect, thank you.
Thomas, wonderful presentation. I think in private conversations, you shared some moment then when you actually came to Iowa State and looking at the different research areas, eventually decided to work on this double haploid and established a facility, created this wonderful platform to training graduate students, doing research. What was the moment you decided to go after this double haploid research area?
It was not a particular moment, but it was, I would say, an evolutionary process. So I came with different themes, and there were different themes in various PhD projects going on related to virus resistance or biorenewables, and I had some work on ryegrass, and it was a bit of an evolutionary process. At the same time, I was trying to get funding, and the areas that I saw promise to obtain external funding and to sustain over a long time, I was continuing ultimately. So it was kind of in waves that there were usually cohorts of a handful maybe of students working on different themes. And the ones where I could see they will be able to get external support ultimately, I was broadening those, whereas others were kind of vanishing over time.
Another short question. Seems like you showed a lot of wonderful research discoveries. Can you summarize the proportion of the times when you have a hypothesis and then you guys started working and it worked and you show the result? How many times it didn't work that the reasoning has, is some flaws?
I didn't keep track of those. Yeah, but it's true. I was wondering, like this slide on the mapping, actually it summarizes the work of 2 PhD studies. And so that's how it gets condensed. But the nice thing here is that it was possible to work on a topic, take it to a certain point, and then it brings up new questions. That's what I like ultimately, that initially we thought the age technology is a really established technology. We just use this inducer from Hohenheim and we use it and do service. And then the problems that we encountered with working with those inducers, the variation in induction rates, problems with doubling, also that variation, that really stimulated us to then look closer and ask questions. And yeah, I really am happy that we could have this sequence of events, and I'm excited about this Haploid frailty idea, where we have a GWAS population in the field this summer to learn more about that. And yeah, I think we have to get started somewhere, but you also need the support. If there's no support, then it's ending after a short time.
There's no more questions. Let's thank Dr. Lübberstedt again.